Good morning, one and all present over here. Today in this class, we will be discussing an organism called Clostridium botulinum. Now, this organism you have commonly heard about in your microbiology class, that Clostridium species, right? Now, these organisms are actually a diverse group of pathogenic bacteria, which is initially grouped by their ability to produce botulinum toxin. So, they are basically a group of pathogenic bacteria, and this particular pathogenic bacteria is basically comprising of, it is being grouped together. So this pathogenic bacteria is being grouped together by their ability to produce a toxin, and this particular toxin is called as your botulinum toxin. Now this Clostridium botulinum basically comprises of 4 groups, 1, 2, 4, and even there are certain other species like Clostridium butyricum, Clostridium berati. These are some of the Clostridium species which also produces botulinum toxin. So this is about the basic introduction of Clostridium botulinum, and you mind it, this particular toxin which is called as botulinum toxin is the deadliest type of toxin, and it is a neurotoxin, and it is as deadly as a snake venom. So next we will move on to the very different type of the character of Clostridium botulinum. Now before we move on to the, we first give you a brief description of Clostridium botulinum. Clostridium botulinum is a gram-positive rod-shaped bacterium. So it is rod-shaped bacterium, and the best known are its neurotoxin. So I have told you that the botulinum toxin is a neurotoxin, and what is happening is that this neurotoxin is further subdivided into type A and G, A, B, C, D, E, F, G. So this is another important thing, and this will ultimately cause the disease botulism, which is your flaccid paralysis. I will rather say flaccid paralysis because it is a muscular paralysis, of course, but it will bring about flaccidness. So C. botulinum is an anaerobic spore former which produce oval-shaped subterminal endospores. So if you draw the bacterium, so if this is the central portion, so subterminal is somewhere over here. It is not terminal endospore. Subterminal endospore is basically present in your Clostridium botulinum. So next we will move on to the character of Clostridium botulinum. So let's talk about the character of Clostridium botulinum. So when I'm talking of character of Clostridium botulinum, this is aerobic bacilli, and this basically is spore forming, toxin, heat sensitive, prefers low acidic environment, cause botulism disease. So next we will move on to the other aspects of your botulism. So this is your botulism microbiology. Toxins is produced by Clostridium botulinum. Now, what is toxin? Toxin which brings about a toxic effect. And you know that bacterial toxin is basically divided into endotoxin, and exotoxin. Now, when I am talking of endotoxin, that means toxin inside the cell. That means toxin is a part of the cell. Exotoxin is a toxin secreted outside the cell. So, bacteria are 0.5 to 0.2 micron width and 1.6 to 22 micron in length. It creates spores that remain dormant for 30 years or more. Spore extremely resistant to environmental stress such as heat, light, etc. So, Clostridium toxin is a exotoxin, and moreover, exotoxin is classified as cytolytic toxin, that is your membrane rupturing toxin, superantigens, which causes a heightening immune response, non-specific immune response, and AB toxin. And usually, this AB toxin is the most prevalent toxin of your exotoxin. So these are the different exotoxins, and botulism is caused by exotoxin. So we will move on to the next other part of your Clostridium botulinum. So let's move on to the toxins. Characteristics of toxin. So liberated during the growth. In which phase? Usually toxin is basically liberated in your exponential phase. And there are 7 types of toxins I have already said, A to G. Now these are antigenic, and when it is antigenic, it is having a light chain and a heavy chain. Inactivated at 100 degrees centigrade for 20 minutes. So 100 degrees centigrade is actually required. And most potential biowarfare agent, and lethal dose is 1 to 2 microgram. So you understand that what type of toxin it is. So next, we will again move on. So if you have any query, any question, you can write into the forum of the Google Classroom. So let's move on to the other aspects of the toxin. So botulism toxin is synthesized as a single polypeptide chain. So it is basically synthesized as a single polypeptide chain. And the toxin, if it is nicked by bacterial protease, it will have a light chain, which is your A fragment, and a heavy chain, which is your B fragment, connected by disulfide bond. A fragment of nicked toxin becomes the most potent toxin found in nature. So this is the most potent type of toxin which is basically found in nature. So next again, the different aspects of your toxin is, so next, you know, the pathogenesis of the Clostridium botulinum. How does it enter? So they basically enter through food and water. We call it feco-oral route. It comes out by the feces and it enters through the oral route. And wound infected with C. botulinum. So if you have wounds infected with the C. botulinum, then again it is being done. Ingestion of C. botulinum by any cause. These all can result. And most important is the canned food, which you cannot forget. In the canned food, sometimes they survive the best and you will have gas in the can. So you have to be very cautious while taking the canned food. The most common contaminated food, vacuum packed or canned alkaline food. Fish, green beans, any home canned food. Food eaten without cooking. That is also one of the important aspects. So now we will move on to the pathogenesis, you know that for normal nerve transmission, we need to have the synapse, we need to have the acetylcholine to be passaged. And you know that in this particular neuron, you have synaptic vesicles. The synaptic vesicle is loaded with the neurotransmitter, and they have a receptor called synaptobrevin. This is the snare protein, synaptobrevin. And onto the membrane there is another protein, that is your syntaxin and SNAP25. So it is the interaction between the snare protein with that of this particular syntaxin and SNAP25 which will ultimately fuse and will release the neurotransmitter at the synapse, and this neurotransmitter will bring about the muscle contraction. So this is the mechanism by which acetylcholine basically is transmitted through the neuron and bring about muscular contraction. But if I say that how does this particular botulinum toxin acts, then you will have to understand, again you have to take into account this particular vesicular fusion by snare. You know that this botulinum toxin, you get to see, has been endocytosed. And once it is being endocytosed, it is having heavy chain and light chain. So the light chain, you get to see, is basically taken out, and they basically cleave the snare protein complex, this particular synaptobrevin, SNAP25, all this. And so the snare vesicular fusion is stopped. And as the vesicular fusion is stopped, neurotransmitter is not released. And as a result, there is no muscular contraction, only muscular relaxation. And that brings about flaccid paralysis. So today, for this class, we have studied till the pathogenesis. So we have studied what is Clostridium, what is its morphology, what it is various characteristics, and its mechanism, pathogenesis. So thank you once again. If you have any question, query, you can communicate in your Google Classroom.